Welcome to the first of our 3 podcasts on the nervous system. And in this first podcast, we are going to primarily focus on neurons, the cellular components of neurons, and also how neurons regenerate. So we're going to understand what neurites are, the dendrites and the axon on a neuron, the cell body, and the different molecular and functional compartments of each of those, and how they relate to neuron function. We're also going to describe axonal transport. Now, this will be something new that we didn't cover in other classes, but we're going to talk about the molecular mechanisms, the hardwiring for the transport, and then directional movement, either retrograde or anterograde. So the nervous system we know from other classes, anatomy and physiology, we can break down into the central nervous system and the peripheral. The central nervous system, the brain and spinal cord, we can have the motor division, which primarily we focus with the motor division on somatic, but we are very familiar with the autonomic nervous system, that being the parasympathetic and the sympathetic divisions. Sympathetic, remember, is the fight or flight, whereas the parasympathetic is the rest and digest system. These are functioning in those non-skeletal muscle regions: smooth muscle, cardiac muscle, glandular secretions, digestive tract. So here we can see those different divisions: sympathetic, parasympathetic, the different innervations. We can see our collateral ganglia, our terminal ganglia. Again, this is just a review of those things we covered in human physiology. The peripheral nervous system, we're going to be comprised of either ascending, afferent components, and these are the sensory neurons that are bringing information to the central nervous system, and the descending or efferent components, which are the motor outputs that are going out to their target tissues. Now, the cells that comprise the functional part, the actual electrical signaling of messages, these are going to be the neurons. And these neurons are going to be arranged in different locations. They're going to have very different shapes, and that's how we're really going to classify neurons as a whole based on their shape. But first, generally, we're going to look at neurons and look at the different compartments and components of a neuron of any particular shape. And certainly neurons are going to have a cell body, which is called a soma or parakaryon. This is where we're going to have our cytoplasmic components and where we're going to find most of the volume of the particular cell. Now, neurons are fairly easy to identify. The nucleus is extremely large, and the nucleolus is going to be very well defined. It's going to look like a huge eyeball in the field that you're looking at in a histological picture. Now, when you look at the rough endoplasmic reticulum and the ribosomes that are present within inside the cell body, when we see those, these are referred to as Nissl bodies, and it's going to be fairly indicative of looking at a neuronal cell. So here we have the diagram. In this, once again, we can see the large nucleus, very prominent nucleolus. It will look like the pupil of an eyeball. Then within the cytoplasm, these different structures that we're going to see very prominent as well in the cytoplasm, this is going to be our Nissl bodies. In an actual micrograph, here you can see our neuron. We have 4 neurons. We can see the large, very round nucleus in 3 of these and the prominent nucleolus. And then all of this dark material that we see in the cytoplasm, those are our Nissl bodies. That's really a light level microscopy term. Now, part of the cell body just at the beginning of the axon is going to be the axon hillock. This is going to be an extremely light staining region, and that's really how we're going to identify the axon hillock and help to distinguish the axon from the dendrites. Remember, the axon hillock, that's the location where we're going to have an extremely high concentrations of sodium voltage gated channels. The sodium voltage gated channels are not located on the cell body or the dendrites. That's why we can only initiate the action potential at this light staining region at the very beginning of the axon. So in this particular neuron, here we can see the soma. Here's our nucleus with a nucleolus. We have a neurite extending in this lighter portion that we see at the very beginning here, also adjacent to the soma. That's going to be our axon hillock. Here again we have another image, cell body, nucleus, nucleolus, many neurites, these processes extending away, and we can't tell which are dendrites and which are axons until we see this light staining region, fairly large neurite extending away. Typically the axon is going to be larger in diameter than the dendrites. So with this being the axon hillock, the axon, pretty convinced that these are going to be the dendrites of this particular neuron. Now, other organelles we see within the neuron, we have a smooth endoplasmic reticulum. Now, these are going to, of course, be the production of lipid and the recycling of membrane phospholipids. However, we are also going to be a place that we possibly sequester calcium. Remember, calcium is going to be used at our terminal boutons, where we are going to allow for the influx of calcium binding to calmodulin, eventually leading to the release of neurotransmitters at a synapse. This is going to be a place where we might sequester and store excess calcium that then we can transfer back and forth to the axon terminals. Certainly we have to have energy, ATP, for a lot of the work we're going to do, certainly for the sodium-potassium pumps we use. So a ton of mitochondria are going to be scattered throughout the cell, but we're going to see a lot of these also at the axon terminals. Now, part of the transport that we're going to talk about toward the end of this podcast is going to be directional movement of certain materials, either vesicles that store materials or entire organelles. And mitochondria are some of these organelles that are going to be transported, and they're transported traveling along a rail system made of microtubules. And this is going to be mediated by certain accessory proteins that we call microtubule-associated proteins, or MAPs. And we'll get into the specifics of those at the end of this podcast. Cytoskeletal components spread throughout a neuron. Certainly microtubules, as we have talked about, used for transport. The intermediate filaments that you find inside of a neuron are simply called neurofilaments. And then, much like we see in other cells associated with the plasma membrane, terminal web, and epithelium, we're going to have the microfilaments comprised of actin in these cells. So this just gives you a little example of sort of the rail system and the cytoskeletal system that is built up within the neuron. You can see to the left, we have our dendrites. And on the right, extending away, we have our axon. And we have this system of microtubules extending throughout the cell and the neurites. And especially in the axon, we are going to have a nice rail system for transporting materials in both directions. Now, as I've said before, neurites, you've heard me use that term, these are all neuronal cell processes, and we can classify them as dendrites or as the usually singular axon. So these are defined as the source of information that's being transmitted toward the cell body, and often this happens through an axodendritic synapse, where we're going to have those neurotransmitters released, binding to the chemically gated channels, seeing shifts in our ions, either an EPSP, an excitatory postsynaptic potential, or an inhibitory postsynaptic potential happening on the dendrites. Now, the dendrites are going to possibly have different organelles than we see, or different components and percentages of organelles, but dendrites will lack the Golgi apparatus. You will not find those present in the dendrites. You will, however, find mitochondria, because once again, as we're changing the distribution and the gradient for our ions through our EP and IPSPs, we're going to have our sodium-potassium pumps that are going to regenerate those gradients after we have the stimulus. Usually dendrites are multiple. You're going to see many of these processes extending away from the cell body, and as we've mentioned before, typically for the axon, you will see a single axon. So most of the neurites you will see will be the dendrites. Here's a silver stain of two neurons, and as we see, there are a number of neurites extending away. I think we can pretty easily pick up this very long dark process. That's going to be our singular axon, and then all of these other processes extending off of the cell body, these are going to be our dendrites. Now, one thing I'd like to point out on the axon, typically in physiology, we pointed this out as just this elongated process that ends in a terminal bouton, the button. But notice in this one, you can have processes extending away from the axon itself. These are called collateral branches. I just wanted to point that out. This is a nice image depicting some of these collateral branches extending off of this axon. So the axon, transmitting information away from the cell body, longer than dendrites typically, larger in diameter typically than we see, and we are going to see that conduction velocity is directly proportional to the diameter of that axon. And really the myelination that occurs enhances that thickness per se and enhances the conduction velocity. Remember, a myelinated axon transmits an impulse over 100 meters per second. In that previous slide, we saw the different collateral branchings that we can have along the extent of the axon. And toward the end, we can have branches at the end that are referred to as the terminal arbor, arbor vitae, tree of life. And as these branch out, we can actually form synapses with multiple target cells and multiple target tissues. It's not just one small button. So here we have a diagram showing, again, the cell body, small, thin processes extending away. Those are going to be our dendrites. Then our one long axon that splits, and we have our terminal portion. You could consider this a collateral branch, this another collateral branch. It's hard to say which one's the axon and which is collateral. We'll just call those both collateral. And then you can see the terminal arbor as we branch out in this location. Now, we mentioned the myelination that occurs, and we will talk about that in a separate podcast, but here you're looking at a transmission electron micrograph. Inside, we have the axon and the cytoplasm of the axon, this Nissl material. And here, what looks like the grooves of an old vinyl record, that is our myelin sheath. And out here, we have the cytoplasm of our myelinating cell. And if this, in fact, is a peripheral nerve, then we know that's going to be a Schwann cell. And one way we can tell that this is a peripheral nerve, as we look at the cross sections of this material outside of the cell. Can anyone venture a guess as to what these are? These are cross sections of collagen fibers, and you're not going to find collagen in the central nervous system, not to this extent. So we are looking at peripheral nervous system, which would make this the cytoplasm of a Schwann cell. Now, axonal transport is very interesting. We're going to use the cytoskeleton and particularly microtubules as this rail type system. And the microtubules are not going to be the static iron rails we think of as much like a train will use, but these are going to be more like the moving sidewalks you would find at an airport terminal. As you're walking through the airport, you have two sides, one moving sidewalk in one direction and the other moving sidewalk in another. Microtubules are constantly being assembled and disassembled. They have a polarity to them. So there's a positive end where you have assembly and a minus end where you have disassembly or breaking apart of the microtubule chain, and that draws that almost as a conveyor belt as you're moving along in the cell. In this particular image, we can see that we've got tubulin plus GDP, which is activated. And then we have tubulin plus GTP. We got to break that down so that we can assemble that into our microtubule chain. So here we can see we are assembling our microtubules. This would be the positive end. This would be the negative end where we disassemble and we lose components of that microtubule. So we're extending in this direction, continually pushing the microtubule in this direction. Well, actually, I'm sorry, if we're disassembling in this end, adding to this end, what we're doing is we are pushing the microtubule in this direction. This diagram gives you an indication of the two-way traffic that is occurring in the microtubules. Not only are the microtubules moving, but certain ones of our MAP or microtubule associated proteins are going to be able to interact with the microtubules in such a way to control the direction of movement. Here we can see that some of our movement is going to be following the pink arrows toward the terminal end of our axon. And then with the black arrows, we're also using microtubules to move back to the cell body. And these are going to be different for different vesicles, different organelles, and we're going to use different molecules to accomplish this type of movement. So once again, we're using microtubule-associated proteins interacting with not only the microtubules, but also binding to their transport packages to move them to or from the cell body. So in this illustration, we've got our microtubule assembled here, and we've got a package that we're transporting. We also have a microtubule here, and we see in this particular instance, this molecule is moving towards the disassembly end of the cell. This molecule is moving towards the assembly side of the cell. And the molecules we're going to be talking with are dynein and kinesin. And you'll have to forgive the spelling here. This looks something like French. I'm not exactly sure. I just really like this illustration for our purposes. So we're going to talk about this type of transport and how dynein and kinesin facilitate these things. So first, we have anterograde transport. And anterograde transport is from the cell body to the axon terminal. And this is going to be fairly fast. You might have 400 millimeters per day as far as the rate of transport that we see. So we're going to be moving organelles, vesicles, macromolecules such as actin and myosin. And we are going to use kinesin, is the microtubule-associated protein that's moving toward the axon terminal on our microtubular system. So kinesin is going to interact with the microtubule. It's going to be able to hang on to those transport products that it's moving toward the axon terminal, but it also has ATPase activity. So as it cleaves its energy, there's going to be a conformational change, which is going to cause kinesin to take 8 nanometer steps per ATP molecule. Once we get to the end, we're going to dissociate that relationship. We're going to lose the package, and it will have been transported to the terminus. Retrograde motion, or going backward, is going to be transporting from the axon terminal to the cell body. And this is only going to go about 200 millimeters per day or 0.2 millimeters per day. It varies widely, but it's slower than we see with the anterograde. Now, in this case, we're transporting small molecules, proteins possibly to be degraded. Our neurotransmitters are being recycled. They're going to get sent back to the cell body. So soluble enzymes, and then that endocytic material, possibly even viruses. This is one way in which viruses affect our neurons and use them. Instead of kinesin, for this type of movement, we're going to use dynein. And again, dynein is going to have ATPase activity. It's going to be able to interact with microtubules as well as anchor and hold on to these transport particles. And as you look at dynein, this is sort of a flip book. If you remember the old flip books, the cartoon flip books, what's happened is they've taken several static rotary shadowing electron microscopic images, and they put those together to look at a dynein molecule. Now, I don't know if you can see it in the middle, but this is almost like a stick figure that has one leg flipping over the next. And it's going to be right in the middle of this image. Here you can see a microtubule. Do you see how the legs are like flipping over and over and over? That gives you sort of an idea of how the utilization of ATP and ATPase activity can cause dynein and kinesin to conformationally change and take these roughly 8 nanometer or so steps all along the length of a microtubule. And that stepping along, again, results in the movement of possibly entire organelles, such as a mitochondria, or small packages of vesicles, either toward the end, which is anterograde, or back toward the cell body, which is retrograde.